Hello and welcome to another video in the lecture series entitled Advanced Biomaterials. So in our previous videos we had seen about polymers. Now what are polymers? Polymers are an interesting class of materials which have been used or which are being used in different types of applications in the industrial as well as in the therapeutic sector. So we had seen how polymers are fabricated, how polymers are used as smart biomaterials as well. So smart polymers are the ones which can show an observable change in response to an externally applied stimuli or stimulus. And because of this, they can be used in fabricating different types of devices such as biosensors and also therapeutic interventions for designing smart materials. So in today's video, we'll be looking at polymeric nanoparticles. Now, what are polymeric nanoparticles? So, polymeric nanoparticles are a class of polymeric nanomaterials which can be in the size range of 1 to 1000 nanometers, and these can be loaded with active compounds entrapped within or surface absorbed onto a polymeric core. So, these polymeric nanoparticles are essentially used as delivery vehicles. So these the delivery vehicles which polymeric nanoparticles are used as, so the active agents can be either entrapped within or surface absorbed onto the polymeric core. Now, the active agent for which these polymeric nanoparticles serve as the delivery vehicle can be of two major types, such as the drugs and genes, that is nucleic acids, such as DNA or RNA. Now, in both of these cases, the polymeric nanoparticles herein are used as therapeutic agents. In this video, we'll be concerned mostly about the therapeutic purposes or the therapeutic applications of such polymeric nanoparticles. But their applications is not only restricted to the therapeutic sector, they are also used in the industrial applications as well, especially in food processing and food packaging industries, and also in other fields of therapeutic and biomedical applications as well. Right? So, these polymeric nanoparticles, based on their construction, can be classified mainly into two types. So based on their shape or construction, they can be as nanospheres or nanocapsules. So, nanospheres are the system wherein the polymeric network is intertwined in a spherical orientation. So, these are the polymeric chains or the polymeric networks. Now, the active agent or the drugs can be surface absorbed onto the surface of surface of the spherical shape which the polymeric nanoparticle assumes, or these can be entrapped inside the meshwork of the polymeric network chain. So it can be entrapped like this. So these are the nanospheres, and these nanospheres are called as matrix systems. Because these polymeric chains essentially comprise a matrix in which these drug molecules or the drug agents or the active compounds can be entrapped or they can be surface absorbed on the polymeric on the spherical shape of which these polymeric chains assume in the form of these nanoparticles. The second construct is nanocapsules. So nanocapsules is basically like a shell which is present and it has a viscous or an oily type of a interstitial space. Now in this system the drug molecules or the active ingredients are present or are dispersed in the interstitial space of these nanocapsule systems. So these will be the drug molecules which will be present inside. Now these systems are known as the reservoir system. So according to the construct, the classification of these polymeric nanoparticles were nanospheres and nanocapsules. Now coming to the main applications of such constructs is that these constructs will be able to protect the active bioingredient or the active biological ingredient inside the harsh in vivo conditions of living beings. So if we are trying to introduce a drug or a gene for therapeutic purpose inside living bodies, these therapeutic agents or these active agents can be subjected to degradation by different enzymes or different physiological processes which happen in the body. These polymeric nanoparticles or these special constructs actually protect the active ingredients from destruction when they are administered in vivo systems. Also, they help in targeting the active ingredient towards the desired location of their location of application of or action. So, the main advantages of these are targeted delivery and protection of active ingredient from degradation. These are all for in vivo applications. Right? Now we will be discussing about two main types of delivery agents. One is for drugs and one is for genes. So for drug delivery applications, these polymeric nanoparticles are constructed as nanospheres or nanocapsules. Now in order to explain it, let me erase out some of these parts. So we have seen for drug delivery systems how these constructs can be. There is another application where these polymeric nanoparticles are used to deliver genes or therapeutic nucleic acids inside the nuclei of target cells. Now, in designing such applications, the consideration is that it has to be targeted or it has to be sent at a specific location, desirable location. So how it is done is that the surface of these polymeric nanoparticles can be conjugated or modified with specific agents of high bioselectivity or high selectivity towards a specific type of tissue. That is, these polymeric chains in the construction of such polymeric nanoparticles can be conjugated with antibodies. Now, these antibodies are very specific to a given tissue type or a given cell type. So, if we are trying to devise or trying to fabricate a delivery system consisting of polymeric nanoparticles with drugs for the treatment of a specific type of cancer, we can coat the surface of the polymeric chain in any of the two types of systems with specific antibodies which can be selectively targeted to that particular cancer cell. Now, when we introduce such surface modified antibodies inside the bloodstream, these will be targeted to that particular cancer type, and the drug will be released at that cancer type, and its its mode of action will be very specific to that particular cancer tissue. And also, there are these constructs which can deliver therapeutic nucleic acids or genes inside the nuclei of target cells. So, for delivery of nucleic acids, these polymers which are used are cationic polymers. That is, polymers having a positive charge. So, these polymers are conjugated with genetic material, that is DNA or RNA, via electrostatic attraction at the phosphate groups. So the phosphate groups CO4 minus have a negative charge, and these cationic polymers will have positive charge. So these polymeric chains having positively charged will interact electrostatically with the negative charged phosphate groups, and this interaction will give rise to a complex which is known as polyplex. So these electrostatic interactions will be stable at the physiological pH. Now these polyplexes or these complexes will then be used for delivery of these nucleic acids inside target cells. So these are the main applications of these polymeric nanoparticles, where they are used as non-viral gene delivery applications. So these were the two main applications of these polymeric nanoparticles in the therapeutic sector. Now let's come to the class of the polymers which are used for the fabrication of polymeric nanoparticles. Again, like any class, these are classified as natural and synthetic. Natural polymers include chitosan, dextran, gelatin, and many others. Synthetic polymers are used because their properties can be tunable based on our desired applications. Such include polyl lysine, polyethylene imine, and many others. Now, there are some factors which can influence these gene transfection efficiency or this gene transfer efficacy. Now also this is dependent on the type of materials used, and also they will be dependent on the structure, molecular weight, and surface charge. So these are the factors which affect the gene delivery efficacy of such polymeric nanoparticles. Now, what are the properties of polymeric nanoparticles, or what are the rationale behind using such polymeric nanoparticles for different delivery applications. So, what are the properties? Number one is biocompatibility. Since these are delivery agents which are directly used inside the living body, these materials first and foremost must be biocompatible. They should not cause any tissue toxicity or cytotoxicity, and they should be compatible inside the body. The second should be biodegradability. Again, since we are using these polymeric nanoparticles for the delivery of certain agents inside the living system, we actually are interested in using materials which will be degraded in the body over time, and they should not produce any toxic byproducts of degradation. So biodegradability is an essential consideration while using polymeric nanoparticles for designing any delivery vehicle. The third one is their facile chemistry. Which is versatility in functionalization. What it means is that their chemistry can be modified based on our particular application for which we are trying to fabricate them. So, functionalization is an essential modification technique of polymeric nanoparticles or any polymeric systems. So, by modifying the functional groups, we can impart new properties in the materials, and we can exploit these properties for a specific type of applications or a specific type of delivery to a given tissue. And finally, the uses the ease of synthesis and scalable production. Compared to different types of drug delivery vehicles, polymeric nanoparticles are very easy to produce. Now, we will be looking at some of the important or very commonly used fabrication techniques of polymeric nanoparticles in our upcoming videos. But comparatively, these polymeric nanoparticles are easy to prepare and easy to load with active agents. And also, their production can be scaled up at industrial levels for achieving a wide scale therapeutic use or therapeutic application. So, with this, we will conclude today's video. So in today's video, we had seen that what are polymeric nanoparticles, what are the types of polymeric nanoparticles which are used in the therapeutic domain for drug delivery or gene delivery, what are the types of polymers which can be used, and what are some of the important features which can influence the gene transfection efficiency of using such polymeric nanoparticles, and finally, what are the properties of such polymeric nanoparticles. So with this, we'll end today's video. Hope you enjoyed it. See you in the next video. Thank you.